So I'm very pleased to welcome everybody here to this session honoring Kate Freeman, who is receiving the William S. and Carelyn Y. Reeburgh Lecture Award. The goal of this lecture is to recognize a prominent scientist who's making an impact in the field of ocean sciences or biogeosciences with novel field or laboratory measurements, and to create opportunities for AGU members to interact with well-known and accomplished scientists and peers by sponsoring an annual lecture at the AGU Fall Meeting. It alternates in every other year between biogeosciences and ocean sciences, with biogeosciences taking responsibility for the odd-numbered years. That's this year. So we're thrilled this year to honor Kate Reeburgh with this lecture. And I'm going to turn this over to Lise Pendle, the president-elect of the section, to say a few words about Kate's being a perfect candidate for this award.
Thanks, Ariel. We're really pleased to honor Kate Freeman this morning. She's been a trailblazer in compound-specific isotope analysis. I've gotten to know Kate a little bit better through the privileged opportunity to co-teach with her in the IsoCamp in Jim Elringer and Terry Serling's isotope course that's been running for over 20 years. And I think Kate really exemplifies the spirit of this award because of her, not only because of her fantastic research achievements, but also because of her wonderful mentoring and inspiration of many, many young scientists from all over the world. So Kate, please.
Thank you.
We have an award to present.
Award.
Oh my gosh. Wow. I hope that gets through security at the airport. All right, am I on? So good morning, everybody. Thank you for coming. I want to start by thanking Ariel and Elise for this really wonderful honor. I also want to thank Bill and Carelyn Reeburgh and just make a comment that I've known Bill's work since I was a student and followed it with great interest, his work on methane in sediments. And I did not have the opportunity to know Carelyn, but I understand she was just a wonderful and very dynamic individual. So today I'm going to share with you a journey that we've been on, that I've been on since my earliest days in science, tracking life's fingerprints, that is looking at carbon isotopes in molecules. And I'm going to highlight today the tools that have made these kinds of measurements possible and how they've evolved. And we'll end with some of the newest and the latest and the exciting research opportunities that are emerging today. Okay, so I study trash. These are just some of my friends. These are biomarkers. Biomarkers are molecular structures that preserve on geologic timescales in near recent sediments or ancient sediments. And I think this points, yes. So you can see here's our trash can. And then if you look down here, these are functionalized compounds that have hydroxyl groups and double bonds. And then under geologic conditions, a lot of that is lost, and you end up with hydrocarbon skeletons, but that still preserve the basic structure of the original biological molecule. And so that's what gives us the concept of a biomarker. It contains the molecular structure that signifies its origin as a biological compound. And my work and the work of my students and the folks I will highlight today is really about taking these kinds of structures and measuring isotopes of the various elements or the various positions, or sorry, of the components of these molecules. And this kind of work actually dates fairly far back, and so we'll start with some of the first kinds of instrumentation that was used to make carbon isotope measurements. This is the publication by McKinney et al. In 1950. This is from the Urey group, and you can see the basic technique that we use to this day is a dual inlet with an isotope ratio mass spectrometer for light elements. And the real breakthrough in this that happened is the advent of differential measurements. So you compare a standard to your unknown, and you can measure that difference very, very precisely, more precisely than you can measure the absolute number. So as long as you know the composition of your standard, you can get a very high precision interpretation of the value for the unknown. And this legacy of differential measurements persists today in the notation that we use, which is a delta notation, where we compare an unknown to a known and then scale it on as basically a parts per thousand. Okay. So one of the watershed breakthroughs in isotope measurements of organic compounds was the advent of continuous flow techniques. This actually, well, the previous slide I showed you dates back 70 years. This one dates back now 40 years. And this is the first publication of, from John Hayes' group with his student Matthews, where they took a gas chromatograph and coupled it to a mass spectrometer using an interface. They were using packed columns in those days, so this interface just sniffs a little bit of the gas flow from the column and then sends a sniff of that into the isotope mass spectrometer. It was a pioneering effort, a visionary effort, and laid the groundwork for the work that I came along and did some years later. I just want to point this out. This is the actual jet separator from that work. I was a student at Indiana, and I was in John's lab, and we were throwing away old stuff, and I encountered this thing, and I knew exactly what it was, and I put it in my pocket, and I have it on my desk to this day. Well, there's John, and my work with John in the mid-late 80s led to the development of capillary techniques for isotope measurements of individual compounds. I love this picture of John. It's actually him at AGU last year, which is the last time most of us saw him. He passed away in January, and we miss him. I miss him personally a great deal, and I think the community does as well. His work with me, or my work with him, I should say more properly, really led to breakthroughs by scaling the technique down to the capillary separation capability, which allowed us to really get good chromatographic separation and then precise isotope measurements. I will say that this work was done in partnership with Martin Scholl, who was at Chevron at the time, and the company you guys know as Thermo Scientific, but in those days was Finnegan MAT. It was a very exciting and exhausting and sometimes very frustrating thesis project, I'll say. So, as I mentioned, John passed away earlier this year, and there was an outpouring of support for an opportunity to honor John, and I'm very pleased to announce a new award that's being sponsored through the Geochemical Society, made possible by the generous contributions of John's former students and his family, and others who knew and embraced him and his work. And so this is an award that is for a mid-career interdisciplinary scientist whose outstanding accomplishments and innovative research lie at the intersection among isotope geochemistry, organic geochemistry, microbial ecology, biogeochemistry, astrobiology, or analytical innovation. These are all the areas where John made amazing contributions. So we don't expect the candidate to do all of those things, but some intersection. So I just will note that this has just been announced, and the application deadline is February 20th of 2018. So moving forward, this is the first sample that we published using the isotope GC methods, or the GC isotope methods, and your eye is probably immediately drawn to this beautiful illustration. It's a reconstruction of the animals, the fauna, and the vegetation surrounding Messel, which became the Messel Shale. It's about 47 million years ago. This is in Germany. It's now a World Heritage Site because of the exquisite preservations of fossils and fossil molecules. And so you see here the data from our first analyses of this, and what really led me to Bill Reeburgh's work is these low values that I've circled in red. These are values that range around minus 60 to minus 75 per mil on the PDB scale, which was stunning to us. The analysis, I had to repeat it several times to convince myself we had it right. And of course, we immediately recognized that these were metabolites or products of organisms that consume methane as a carbon substrate. So we were able to track methane consumption into the individual compounds with these very, very depleted signatures that's characteristic of methane. And this approach to tracking methane continues to be a major tool in methane biogeochemical studies to this day. The other thing to note in here is that there's a few intermediate values around minus 40, and then a lot of values sort of at the minus 20 to minus 30. So it's sort of primary productivity coming in here. I interpreted these in this paper as the organisms that lived at the chemocline and were consuming respired carbon, and then these methane cycling compounds down here are related to methane cycling. Moving out from there, the application of compound-specific measurements has embraced all kinds of ways of making a living by microorganisms. This is from a nice paper by Chris House and his former student Audrey Zirkel, where they've mapped out both the substrate of CO2 and methane, as well as the different kinds of metabolisms that use those substrates and their range of isotopic characteristics. And so we use natural isotope signatures as a tracer or as a way to unpack microbial carbon cycling and indeed plant systems, terrestrial systems as well. I just will make a little advertisement for compound-specific applications that are outside of geochemistry. They are widely used today, and in fact, all major international sporting events have an isotope lab associated with them, and includes many kinds of isotope analysis, but and amongst those is the compound-specific techniques. And so it's widely used in drug tests and forensic science. And also used, and this is actually some of the earliest work in this technique was related to food and flavor authenticity, particularly in Europe, which has more stringent rules about what goes in your food than other parts of the world. And so there's a lot of excellent chemistry and biochemistry that goes on in those disciplines in European laboratories. Petroleum and gas was, of course, an original sponsor of the work and continues to be an important tool in research in those industries. Atmospheric and ocean sciences, it's used to trace carbon. Climate and paleoclimate. My own work has often focused on paleoclimate applications of this. It's used, as I mentioned, in the Messel Shale example, to study biogeochemistry and ecology, Earth history. It's been applied to meteorites, which give you great range in isotope values. It's very dramatic. And then it's increasingly used in astrobiology. In today's presentation, I'm going to talk a little bit about, let's see, a couple of these things. I'm going to touch on some Earth history. I'm going to touch on some climate and paleoclimate. And I'm going to touch a little bit on meteorites and astrobiology. So, my work on paleoclimate has crawled out of the ocean and onto land in the last decade or so, and I've been really fascinated by soils and the residual of organic carbon that's in the soils. This is a picture of the Bighorn Basin in Wyoming, the United States. It's a beautiful place to do field work, and you can see these stacked paleosols, and that red color as an organic geochemist means that there's not much there, and then that is in fact the case. You can see that in this profile here. The curve on the right is the quantity of organic carbon in these soils, and the left is the bulk carbon isotope value. These are spanning what's called the Paleocene-Eocene thermal maximum, a hyperthermal interval of climate that got very warm very fast, and was associated with a fairly dramatic 13C isotope excursion, which we can trace into all manner of carbon in the geologic record. And in this realm here, you see this sort of loss or lacking of organic carbon. That makes our work challenging. But there are many sites where we can get isotope measurements on individual compounds, and we can use that to then unpack the response of terrestrial ecosystems to the warming event from a isotopic perspective. And one of the things that we noticed early on is that when we do this, we can measure plant waxes, which have an isotope value that's always depleted relative to the leaves, which is always depleted relative to carbon dioxide that plants consume during photosynthesis. And we notice that the values of the leaves are more depleted than the values of organic matter preserved in the soil. And this has been a source of trying to understand why this is so different than that we've been working on for the last few years. And we can show that this 13C signal in the bulk is actually modified or attenuated by enhanced degradation of the organics during this time period and the mixing in of fossil carbon weathered from other regions in the basin. So there's sort of fossil carbon component to this and an enhanced degradation. And all of that we were able to unlock using these molecular techniques. But the low organic carbon during that really important interval actually has posed a real challenge for us, and that is that the concentration is so low of the alkanes, they drop about threefold into that interval, and this is for a core. And with a core, you're limited in the size of sample that you have, and so we want that lovely continuous record, but we're challenged because we need big hunking samples to make our measurements. And so we don't actually have isotope measurements in the key interval where that yellow area is because the sample size is too small. So we would need about 650 grams of rock in order to make measurements on those with conventional technology. And so this posed an analytical challenge for us, which I like to think of analytical challenges as an opportunity. And so I'll talk about our work with Pico CSIA, which really tries to lower the sample size requirements for these measurements. I'll talk a little bit about NanoEA, which does the same for elemental analysis methods. And then I'll end with some comments on these new techniques for position-specific measurements. Oh, and I don't know if anyone, I'm dating myself, but this image on the right is of Tinker Toys. So I don't know if you had Tinker Toys as a child or if you had your children play with Tinker Toys, but I spent a lot of time playing with Tinker Toys, and I kind of see myself as still basically playing, tinkering with things. So I celebrate that. Okay, so in gas chromatography, there's kind of a constraint on what you can do that's driven by the dynamics and the physical properties of getting gases and materials through a capillary column. For isotope measurements, we need fairly large samples, at least by the conventional means, and so that means that we sacrifice either a separation power or we have to do things at a really slow pace. And this sort of triangle of constraint means you only can pick 2 out of the 3 on here. But if you look at it another way, well, we don't, excuse me, we're limited by sample sizes, so we'd actually like to go to small samples. So maybe we can turn this challenge into an asset. And that's exactly what happens when you use what are called fast GC techniques, and they really aren't anything fancy except an exceedingly narrow column. So we go down to a 1/10 of a millimeter in diameter, which creates sort of challenges in terms of the pressure regime to get gas through it, but nothing that's not manageable. And the upshot of it is that you move from your conventional technique, which is the red line, to a very sharp, very narrow peak with the fast techniques. So we get high separation power, which is a great benefit. It's very fast because the peaks are sharp and narrow, but which requires faster electronics than is conventionally provided. But one of the upshots here, one of the positives, is that most of the material you're measuring is up in a high signal-to-noise part of the measurement. And it gives us those very small samples. So we've been involved in a partnership with Thermo Scientific and Pratica Polisar at Lamont on developing techniques to make these measurements, and I'm very happy to say that we've made some progress. So here's a conventional GC chromatogram where each of these little pickets represents an individual alkane that's been separated in time, and the vertical is amplitude. And each of these little peaks of alkane gets sent off to a combustion reactor, turned to CO2, and the isotopes are measured. Well, that's how we normally do things, but now that we have Pico CSIA, this is what it looks like. So that entire standard is now all happening here, and each of those peaks is a little hard to see, and I apologize that it's faint, but each one of those peaks is now preserved, but they're half a second or less than half a second wide at half height. And so we've now cut our analysis time down by an order of magnitude. We've enhanced the separation power, and we've moved down to much smaller sample sizes. So that's kind of the showpiece of it. This is the nuts and bolts. Here's the instrumentation. Here's the real genius behind it is Ali Baczynski, who's been working on this project with me the last few years. Pico CSIA now requires 7 times less carbon for the same peak height. That's what's shown in this diagram. It's basically a similar model to what we do conventionally, just everything's narrower, and that means including some of the valve switching or the flow switching is done with microfluidic techniques, and then the other big modification is the electronics are much faster. And then here's some of the results. Here's that narrow peak shown in the middle. That's the Pico peak, and then the conventional peak is shown around it. And here's some precision data that you can see, which is precision and accuracy data showing in picomoles of carbon. And I just want you to notice that this is 50 picomoles of carbon, and that we're getting pretty good precision down even well below that. When we are combusting peaks in conventional or in operational mode, we find that we need about a little less than 100 picomoles of carbon to do this. So between 50 and 100 picomoles of carbon gives us the kind of precision we need for our studies. So I'm very pleased to reveal this to you. We just have submitted the manuscript, and hopefully the review process will help us make it better, and we can get it out sooner. Pico CSIA with this new development, Ali is now running as we speak. She's not here because she's running N-alkanes from that core slide that I showed you earlier. I hope to learn what secrets it holds for us. So that's now possible. And then we're getting kind of crazy ambitions to do all sorts of things, such as refractory compounds like the PAH that's shown here, coronine, which is from the work of Elizabeth Dennis, that, like the alkanes, just kind of disappears during the PETM. So we're really opening, I think, a new analytical window to look at these weathered residual materials. And then the other ambition that we have with this, which hopefully I'll come back next year and maybe share at AGU, is the ability to run biomarkers in very ancient samples. So these are some calculations for Proterozoic biomarkers, where we're estimating that we would be able to have about a 10 picomole of one of these compounds in a gram of rock. So, we need 10 grams of rock, that's doable, to make measurements on these very important markers of our earliest inhabitants on Earth. Okay, let me grab my water. And I'm going to switch from pico to nano. And nano is the name we give to an isotope ratio mass spectrometer with an EA on it, but it's been modified to sample nanomolar quantities rather than the usual micromolar quantities of material. So on the left is sort of a little block diagram of a conventional EA, and then what we do is sort of modify the end gas flow from this and trap the peaks and then re-release them in a capillary configuration. This is basically nothing much different than what Brian Fry did with his early papers on trapping EA peaks, but we do it in a continuous flow environment, and we're able to do it with capillary connections. And I want to highlight the folks down here. None of this, of course, could happen without the wonderful folks I get to work with at Penn State, including Pradika Polisar, Jamie Fulton, Courtney Turek, and Chris Junium, who really were the ones who rolled up their sleeves and made this project happen. It takes a village with this one. And so here's some of the, we published this a few years ago now, but we're using it routinely to this day. Here's a sharp peak of nitrogen. You can see the CO2 is down here. It's equally sharp. We're actually monitoring mass 28 in this particular image, and so that's just the CO fragment of the CO2. But during a measurement, we would peak hop to mass 44 and get a good measurement on that CO2. And there you can see this trapping configuration on the left there. And then, as I said, this takes us from micromoles to nanomoles. What we were interested in, and we continue to be interested in, is isolating compounds or measuring isotopes on compounds that are too big to go through a gas chromatograph. And so we use HPLC techniques. This is illustrating that with a two-dimensional separation scheme for a porphyrin compound. This is from Chris Junium and Jamie Fulton's work. And so we do very careful separation of a peak, and then we re-chromatograph it to make sure it's super clean, and we can check that with mass spectrometry. One of the applications I want to share with you is the more recent one. It's the work of Laurence Bird, who is now a postdoc at Caltech. And he was following the fate or the carbon isotope signals of F430, which brings us back to Bill Reeburgh. This is a gateway for methane biogeochemistry. This is a coenzyme that's long been recognized as the important last step in methanogenesis. And also it turns out that in the reverse process of reverse methanogenesis or methane oxidation by archaea, it is the gateway that brings in the methane. And so we were interested in being able to unpack the sort of isotopic heterogeneity within the cellular biochemistry, because there's some hypotheses out there that maybe CO2 is also consumed along with the methane, and we might expect to see a range of values in the individual biochemical products. And that is what we went looking for. This is a work in collaboration with Kath Dawson, who's at Rutgers now, and Victoria Orphan at Caltech. And this is in Hydrate Ridge, which I don't know if you can see this, that the dot is in the water on the ridge, and then the coastal area of the Pacific Northwest is just to the right of that. The concentration of F430 maps very nicely, or tracks very closely, the concentration of the numbers of aggregate clusters that show AOM activity or methane oxidation communities. And so we have a pretty strong confidence that this compound is associated with methane oxidation. We measure by nano-EA the isotope value of the F430, and we compare it to GC measurements of the isotopes of the archaeal lipids. And what you see immediately here is that there's a real big difference. This is quite a large scale of isotopically. We see different ranges, different settings, and under different experimental conditions when we do some incubations. But nonetheless, it's showing us, so in this particular example, a fairly large difference in the isotope values between these two. And that's led us to think about the substrates, as I mentioned, CO2 versus methane. Some of these measurements, I just want to say, Laurence worked really hard on these, and these are kind of heroic measurements. This is a tricky compound to work with. The organic chemists in the room will note that there's a whole suite of carboxyl groups around the outside of that compound. It's a challenge to work with. So I just want to give a shout out to Laurence for his intestinal fortitude on this project. It was hard. But it's been fun now that we have the data to think about what it means. This is a figure from Laurence's manuscript where we're tracking in blue the methane carbon and in red a carboxyl carbon or from DIC in the environment. And when we look through sort of the pathways that lead to the synthesis of F430, we estimate from this kind of accounting and also from our mass balance of our measurements that the F430 reflects about 60 to 75% carbon from CO2, with the rest coming from methane or an unknown source. And then the AOM lipids are built mostly from methane, and that's really consistent with their sort of charismatically light or low values that are seen in many, many environments. So I just want to say that this really inspires me to think about following biochemistry by looking at isotope values or patterns within molecules, not just between molecules. Everything I've showed you before up till now is between molecules. And now we really want to think hard about what we can unlock and learn from the patterns within compounds. And in this particular illustration, we're showing two different sources of carbon that are tracked within the same molecule. But in addition, there are isotope sensitivities to some of these reactions, and so we may be able to learn about carbon flow or branch points in the biochemistry by looking at individual compounds, even if they come from the same substrate. And so the next few slides I want to share is really sort of highlighting ways that people are thinking about intramolecular isotope measurements. And to do that, we need a little bit of vocabulary, so I'll start with this slide. Intramolecular patterns can be characterized as isotopomers, where you have the same structure and the same mass, but the isotope substitution is just moved from one position to another. So that's an isotopomer. It's like an isomer, but an isotopomer. And that's a subcategory within a larger class of homologs of compounds that have the exact same structure, but different substitutions of isotopes within them. And that's one example. All four of these are isotopologs. These two are a special case called an isotopomer. And that language is now really making its way out into the geochemical literature. And I don't know about you, but I find it confusing. So I wanted to give you, you can't tell the players without a scorecard. There it is. All right, so this is from a figure from John Eiler's paper a few years ago now, where he's really celebrating the possibilities in these compounds. And the previous slide just showed sort of two positions. And of course, in an organic molecule, we have many, many positions, and we have both carbon and hydrogen, and in some cases, nitrogen or oxygen. So there's a lot of dimensions of informational space here that's possible with, as we move towards these measurements. And some are single substitutions, some are position-specific differences, some are mixed, and then I think there's somewhere in here is the clumped. Where's the clumped? These are adjacent samples, so that would be a clumped example, and these are differences but not necessarily adjacent, so they're not sharing a bond, so we wouldn't technically call that a clumped difference. So there's a whole lot of vocabulary that I'm still learning, and I think we're sort of working forward, going forward, we'll all be learning this to some degree. One of the earliest methods to do this came from pyrolysis methods, and this was first introduced by Tom Brenna and his students back in the 90s, and it lay fallow for a little while. We picked up and worked on it with Bob Dias' thesis, and I'll show you some of his data in the next slide. And then it's really been moved forward by a Japanese team and also with Alexis Gilbert, who now works in Tokyo, where they've gone and not only developed this method so it's like reliable, but they actually are now applying it to all kinds of compounds in exciting ways. So I'll just highlight the possibilities here with this work. This is old work now, but I think given my renewed interest in these intramolecular patterns, I've been thinking a lot about it. This is Bob Dias, this is his thesis work, and he was looking at organic acids, which have an alkyl portion shown here in acetic acid by the methyl carbon, and then a carboxyl carbon. And we were able to measure the whole molecule and then compare that measurement to just the carboxyl carbon. And by difference, then you can calculate the alkyl carbon composition. And this was inspired by some work that we had done in collaboration with Steve Franks, who was then at ARCO and was interested in some oil field brines who had some interesting patterns. And so we did some simulation experiments with Mike Lewin at the US Geological Survey. And Bob found that the isotope patterns of the low molecular weight acids in each case approached the composition composition of the inorganic carbon in the reaction vessel. And this led us to speculate that there might be something happening with that carboxyl carbon. And what we found and were able to show is that that carboxyl carbon is in fact exchanging with the inorganic carbon in the aqueous environment. And that's shown here. There's the whole molecule measurement for one of these experiments. There's the carboxyl carbon measurement for those same compounds. And then by difference, there's the alkyl or the hydrocarbon part of the molecule. And so that carboxyl carbon has an isotopic composition very consistent with an inorganic carbon source. We did some work with Jim Kubicki and his student Philippe on making some models, calculations of possible mechanisms for that, and that work nicely showed there's a malic acid intermediate that likely is at play here. And then finally, I just want to highlight that this is not, even though we did this work a while ago, it still has new relevance, and this is some data on the Tagish Lake meteorite and also the Murchison meteorite by Hurd and others that was published now a few years ago that shows that same pattern of isotopic changes with the chain length, and they in that paper use our work to invoke an influence of exchange with inorganic carbon during a hydrothermal phase of the history of these meteorites. Okay. Our colleagues in Japan have really moved this much further in looking at organic compounds in biochemical systems. They published a really nice study on vinegar, the same molecule I showed you in the previous slide, the acetic acid. And in that study, this is the whole compound versus the specific positions, and you can see in C3 derived fermentation products, or vinegar that's been fermented from C3 sources, the carboxyl carbon has very similar isotopic compositions to the methyl carbon, but there's quite a distinction between the two sites in fermentation using C4 or CAM sources like pineapple. So they're interested in this from a biochemical and sort of food authenticity perspective, but it also, to me, shows that even if you don't know the absolute or the isotope value of the whole molecule, the difference itself tells you the biochemistry, right? And I think that's really exciting. Now, it's not news to the folks in France. They've been doing measurements using NMR techniques. This is at Nantes. And they have a whole airplane hangar-sized lab. It's kind of like the size of this room, where they have NMRs that are running round the clock, making very careful measurements, particularly of hydrogen, but more recently they've added carbon to their matrix. And they're using this to distinguish synthetic from natural or other sources, for example, of lignin. This is the maybe most well-known example. These measurements are all just of that little methoxyl group there on the lignin, the vanilla structure. And of course, if you ever go to the grocery store and you are going to make a pumpkin pie, like some of us just did a few weeks ago, actually making pumpkin pies is part of my marriage contract. My husband expects them on Thanksgiving. I don't know if you have similar experiences, but anyway, I digress. And the price differential between the natural vanilla and the synthetic vanilla is staggering, right? Well, here's the secret. It's the same molecule. It's the exact same molecule. And the only way you can tell the difference is by these kinds of tracer techniques. So the price differential can really help us understand why it's so important to be able to make these from a commercial perspective. Okay, so now you're facing an existential choice the next time you do your baking. Are you going to get synthetic or natural? And I encourage you to think carefully about that. But it doesn't stop there. Some of these measurements are even finding greater differences. This is caffeine. Some of you may have encountered caffeine this morning. I don't know. I had my share. And caffeine has ring compound or sites, and then it has methyl sites around the outside. And the ring sites have an isotopic pattern that's really quite different than the methyl sites. And it's big. It's over 50 per mil. So again, there's really dramatic differences that are sort of lurking and waiting for us to uncover them. Now, the food and flavor industry folks are not limited by sample sizes in the same way that we are, and so these techniques are really moving in that direction of making measurements on smaller materials, but they're certainly not there yet. There's some excellent progress, but there's still challenges. So the last thing I want to introduce to you is a new technique that is just emerging. This is from a paper by John Eiler. It's actually in press, I think. It's introducing using an Orbitrap mass spectrometer for isotope ratio measurements. Orbitrap instruments have very, very high mass resolution, and they can also operate at a wide mass range. This particular design instrument has a quad that can allow you to filter out and identify exactly what it is you want to measure. You can send it off into this end over here and collide it with some things and break it up a bit if you care to. And then all the ions sort of hang out here in the C-trap. That's where the cool kids are. And then they can be sent very precisely into the Orbitrap analyzer itself. And the Orbitrap measurement is a kind of a beautiful thing. It makes the measurement of the mass by the cycles, the frequency as the ions rotate around a spindle, and they actually go up and down that spindle, and it's the frequency at which they go up and down that is the mass separation. The separation power, as John highlights in his paper, is enough so that you can identify a fragment that has a 13C, a deuterium, or an extra hydrogen. And it's an amazing separation capability right in the mass range where a lot of our compound or their fragments of compounds would be of great interest. And so this high mass accuracy, this wide mass range, and this high mass resolution is something that John Eiler has been really exploring in exciting ways about making intramolecular measurements. And the way that would work is that you would fragment your molecule in the way that normal organic mass spectrometry works, but then you would zero in on those fragments that contain the rare isotopes. And you can monitor those, you can separate them out, and you can monitor them and get, he's now reaching per mil precision at individual sites. And it's a very exciting, although not an easy measurement. I don't want to lead you to believe that this is commercially available or anything like that. It's still very much on the vanguard. So that leads us to just a couple of summary closing slides. As we think about this frontier of position-specific isotope measurements, we can think about each of these sites as having its own history of a reactant and a branching and products. And with that process of chemistry or biochemistry, there's usually a mass sensitivity. And this, to me, this has always been the greatest limitation in molecular isotope measurements, is that we can measure the individual product with incredible precision, even before some of these techniques came along. But we are always limited by interpreting either the source or the nature of the reaction or the flow of material through that branch point, as illustrated by sort of a classic open system diagram where your material comes in and your product is depleted relative to its substrate, and if all the substrate is consumed, then you end up with a value where you started. And so understanding the extent of reaction is as important as understanding the mass sensitivity. And we basically lack that for most of our paleo applications, and we infer it or we make some educated judgments about it, but the possibility is that with each of these sites will be carrying their own history of a source and process. And that, I think, opens another dimension to help us interpret the origins and the fates of compounds in new ways. So some of the applications that I'm interested in and I could see being particularly exciting include abiotic processes, including thermal reactions. And this actually is an area that's already well underway with looking at clumped isotopes in methane, and we do accept methane as an organic compound. It's only got one carbon, but we'll take it. And so that counts. And thermal processes imprinting on methane isotopes, isotopomers and isotopologs is a very exciting area right now in natural environments, particularly related to energy production, but it's more recently moving into microbial systems and understanding biodegradation and production of methane. Hydrothermal processes I highlighted with Bob Dias's work, and so we can get exchange at specific sites within compounds from abiotic processes, such as the warm aqueous environment. And an area that I'm particularly interested in as well is the interactions of organics with minerals, because I think that's so critical to their survival on geologic timescales or on other planetary materials or meteorites. And so understanding how they're interacting with organics are interacting with minerals, and how that imparts or doesn't impart an isotope pattern at the individual site, I think will be very important. And then, of course, there's a huge array of biotic possibilities, and the two categories are really whether or not there's more than one source of carbon to the biological system, and then the branch points and the synthetic pathways and the mass sensitivities of each of those steps. So I think of it as this triangle. I like triangles. We have biotic processes and abiotic processes, and each of these are imprinted by environmental conditions. I'll just close with an image of Tom Haring, who advised me when I did my undergraduate thesis at the Carnegie Geophysical Lab. Tom, he taught me a lot of things. When we were working in the lab, he would often say to me, shoot first, ask questions later, which was about the antithesis of working with John Hayes, who wanted everything mapped out ahead of time before we did anything. So it was good to have those two different mentors in my life. It helped me have some balance. He was sort of famous for this triangle, and any new analytical endeavor or scientific endeavor, you can only pick two. It can be fast and cheap, but it won't be any good. It can be good and cheap, but you won't be able to do it very quickly. Or you can do it good and fast, but it won't be cheap, right? So, I guess some words of advice from a generation, our predecessors in the science. Tom also was the first to do intramolecular isotope measurements with his very famous paper with Phil Abelson in 1961, where they measured carboxyl carbon and other sites within amino acids. That work is a thing of beauty, and it also was probably not fast because they had to separate each molecule out by hand. All right, so I'll end by saying that studies of molecular isotopes is a field that is full of analytical challenges, opportunities. And I encourage you, especially the young people in the audience, to think about challenges as opportunities, and that these open up new views of carbon and its history on Earth and beyond. Thank you very much.
So we have plenty of time for questions.
Carrie.
Can you repeat the question?
So the question was my thoughts on how to use clumped isotopes to explore organic mineral interactions. Well, first of all, exploring organic mineral interactions is hard, right? Because there's a sort of a partitioning problem there. But what we're particularly interested in is are there reactive sites that play a more important role in connecting to the mineral surface than other parts of the compound? And in soil science, it's well known that amphiphilic compounds will have a polar head group and a nonpolar part of the molecule. The polar head group will interact with the mineral surface, and the nonpolar part sort of dangles out in solution. And then it itself creates an environment where other organic compounds can come in and sort of escape the aqueous phase, if you will. And so it's that difference in the reactivity or sensitivity to the mineral surface is hidden in the sort of electron density of the different sites. And I think that's the possibility where we might find this. My suspicion is it's probably more theatrical with hydrogen than carbon. And that's, I think, where we're starting. It's a good question.
There's a microphone up here as well.
Okay, that's a very interesting talk. I'm just curious, you mentioned that there will be a challenge to interpret these intramolecular carbon isotopes. And so I was wondering if you could offer some recommendations for how to go forward about to go interpreting or these records.
Yeah. I would say it's really, the question is how do we go about interpreting these things? And I would say we're really at a frontier moment in the science, that there's a lot that we don't understand or even know because we haven't measured it yet. And I think we will go hand in hand as we move forward, measuring natural materials, perhaps geochemical materials, but also looking at biological systems or experimental conditions in the laboratory in order to build up that knowledge base in order to go forward. And at least that's what I would like to do. Oh, right. So the question is whether or not nano EA is commercially available. That decision is not up to me, but I will say that there's been one built at UC Santa Cruz. There's I think Syracuse, I don't know if Chris Junium is here, but he's been building one. Jamie Fulton, I don't know if you're here, but he's thinking about building one at Rice. Trinity, Dublin has one. So there are a few sort of home built, and we consult with people as they want to do this, so I'm happy to do that. But I also am now working with Thermo Scientific, and they've given us their new EA design, and we're going to work with them in partnership over the next year or two to try and develop advance the possibilities of commercial applications. Ours is, you know, it's got duct tape and binding twine, and you know, it's kind of a home-built model, so it's not a polished thing, but we love it. So.
Hi. So I was wondering, for Abiotic reactions, we need this larger constraint on isotopic signals.
Yeah.
So maybe if you had any ideas for that, or maybe also your ideas about converting, maybe using like a Urey-Miller system or something where we're making a more standardized isotope measurements.
I'm not sure I got that question. Can you repeat it? It's hard for me to hear because the speakers are going out.
Yes. So first it was just constraining abiotic isotope signals, right? And the second is maybe having a more standardized approach instead of per mil, like a Urey-Miller. Yeah.
Yeah. So you're asking about how do we compare data with now this sort of added dimension of complexity. And I was at a workshop last October, not this year, but the year before, in 2016, where they're really beginning to have an international conversation about what's the nomenclature, what are the standards for position-specific isotope measurements. And one thing I think we really desperately need is a concerted, coordinated effort to create a set of authentic standards for comparison, and that might involve just initially making measurements on the same compounds by NMR, by pyrolysis, and by Orbitrap techniques, just to compare how we do. We're at really early stages in getting those 3 different techniques to talk to each other, or the people who run them.
So I've got a question picking up on one of the previous ones. So to unravel these new isotope effects, right, these intramolecular effects and things like that, requires collaborations between analysts, analytical geochemists, and others who understand the systems that are being studied. So what kind of collaborations do you think should be prioritized, and are they happening? And if not, what can be done to facilitate them?
Yes. Well, I'm very excited to build collaborations. We recently proposed for NII to have a center focused on these kind of collaborations. I think it would involve analytical collaboration because of the different techniques and the challenges of making these measurements in the first place, and comparing the results that we get, make sure we're all measuring the same thing. I think there needs to be biological studies in the laboratory, and then eventually in more complex systems in the field, and then inorganic or non-biological experimentations, and perhaps some clever choices of abiotic source samples in nature. And then modeling, we don't want to leave that off. And so what we'd really like to move forward on is having a predictive capability, right? That's ultimately what we want.
So are we as a community organizing that way? I mean, I know you probably put that in your NAI proposal.
It's in my proposal,
But more broadly, right?
No, I don't think we are. And partly because it's, the NMR techniques are being used by food and flavor chemists. The pyrolysis work has sort of focused on small molecules, and then the people like me who do geologically preserved materials have not been able to really take advantage of those techniques, either by sample limitation or size of the molecule. And so now with the Orbitrap technique, if that can come into fruition in the ways that I think it might, we have that possibility of sort of joining that conversation, bringing the geochemists on board.
Are there any other questions? Oh, one more.
Got another one, yes. Right, so that's a great question. He's asking if you have a substrate and then it's brought into a metabolism, there's fractionation during that metabolism, and there's branch points and so on. And that's really where you need controlled experimental studies to look at the same substrate but different types of reaction or growth conditions, for example. Alex Sessions is actually, he's not here today, but he was here earlier this week. He's very interested in this for amino acids, and he's begun some studies doing that kind of work.
Well, if there aren't any other questions, then please join us in thanking Kate Freeman and congratulating her again for this award.
Thank you, everyone.